I think we set up our reactions the 0.5 way in the 15 microliters. This one must have had less loaded. This is quite a bit older. That one, I think just here I was trying to get you to put loads in there, and it really wasn't necessary.
Okay.
I know why I was thinking the wrong way. I don't usually work with plasmas. I usually work with PCR fragments, and so we never have enough. We always load everything we have. It's really not necessary when you've got gobs of plasmid DNA. So I'll try to learn for the future. All right, nucleotides. Just reviewing what we talked about the last time, which I guess was Monday. We talked about the nucleotide monomer. You should recognize that structure, at least be able to sketch the sugar ribose portion. Probably indicate the 3' hydroxyl, because that's important. Some kind of a base, maybe without any details at all, would be expected to be hanging off the first carbon, and on the 5th carbon we would expect a phosphate or 2 or 3. That general structure, maybe without all the chemical details, is something that I would want you to be able to sketch quick or to recognize. The bases, again, this is too much detail, but I do expect you to know that adenine and guanine are big, maybe twice as big as the single-ring pyrimidines. So purine and pyrimidine, those words are useful to know, that one is big, one is little. It's good to know that RNA uses the U base in place of the T base. So if you see a chain of nucleotides and you see T's in there, it's probably a DNA chain they're showing you. If you see U's instead, it must be an RNA chain. Sometimes in a chain you can kind of peek at the 2 carbon and see if there's an oxygen there or not. I don't think they kind of seem to have left it out this time. Maybe they were trying to be ambiguous. They also haven't given us a base to decide which it is. Here we can definitely tell this is DNA. We see T's, we see double-stranded. We really do expect DNA to be double-stranded. 2 anti-parallel backbones. Bases in the middle, 1 purine, 1 pyrimidine, sticking together by hydrogen bonds only, so easily separated. GC3AT2. All of that stuff seems to be apparent in this diagram. That's all the stuff that really is important about DNA structure. Then we talked about restriction endonucleases, and there was a ton of things we were supposed to pick up about this topic. Number 1, the way they're named, because we're not going to expose you to all 400 names. They're going to be named in this style. The fact that they tend to cut within short 4-base or 6-base or sometimes 8-base palindromic sequences. Not always, but most of the time. So that's kind of something that we expect. They may cut and leave blunt ends. They may cut and leave staggered ends like these. It doesn't make a bit of a difference if you're quite far away. If I've got a double-stranded DNA and then I cut it, I really don't care if my middle finger's longer than my pointer finger. I cut it into 2 pieces. When we go to clone something later, we might care if there's a few hydrogen bonds in there. But it won't be many. If we cut this backbone and we cut this backbone, that's 2, 4, 6, 8 hydrogen bonds. That's going to fall apart. Just random movement in the watery solution is going to yank those 2 apart. We were able to answer a question like this: what is the average fragment size produced by EcoR1, which has a target site of 6 bases? And we did the rough and easy. 1/4 to the 6th gives us a probability of 1 in 4,000. So we expect an average fragment size of about 4,000, and that's kind of what we saw in our laboratory. What is the average fragment size produced by ALU, which has only a 4-base target? And again, 1/4, because we don't know a better number. We could use a more exact number if we knew it. To the 4th power, because there's 4 bases, gives us 1 in 256. That's the odds of finding that sequence randomly if the abundance of the bases was all the same. It's also the average fragment size would be about the inverse of that, 250 bases or so. What is the average fragment size? Hindi 2 is different. It's got that weird pi-poo thing. We'll see another weird one during the lecture today as well. So the calculation's a little different. Sometimes we save this for the test so you don't just spit out 4,000 or 250, because we talked about those. You'd have to do a fresh calculation for this. 1/4 for the G, 1/4 for the T, 1/2 for the pi, 1/2 for the poo, 1/4 for the A, 1/4 for the C. Multiply that together and I think it comes out to about 1 in 1,000. So it is a sort of different number. And I don't know how else you could ask the question, really. If you digest a plasmid of 4,400 bases with EcoR1, how many fragments do you expect would be produced? And I'm thinking just one. I think the odds of it cutting are about 1 in 4,000. I've got 4,000 bases. I'm just, am I going to win this lottery? Yeah, I'm going to win it. One time and one time only is my guess. Is that the right answer? I don't know, but it's an answer. All of those different fragment sizes that we might expect or predict, we could do the digest, run the gel, and with a size standard, make a good, educated analysis of what exactly are the fragment sizes that we produce. Again, we expect the high bands to be brighter. We would expect to add these up to get the total molecule we began with. We've already seen some exceptions to that. A partial band might be kind of high, but it might be kind of light if it was gradually going away. I showed you an experiment in lab the other night where we added up all the fragments and they didn't add up to the total known size of the plasmid. A few went missing and we wondered where they were. So you can think a little bit beyond what am I supposed to get? Does this add up to my expectations, or is there something wrong? This is an important question. I think we talked about this a little bit yesterday or in lab when we looked at those slides, but this is a good question to think about. If you digest human genomic DNA, and it really is the elephant in the room. It's the huge DNA that the whole field of molecular biology was invented in order to understand. So 3 times 10 to the 9th is like the Holy Grail. This is the genome that we're trying to contend with. And this is my calculator. 3. Oh, 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 oh. There's 3 times 10 to the 9th bases. If I digest that with EcoR1, and I'm expecting a 4,000 base pair fragment on average, I divide that by 4,000, 4,000, I get 750,000. So almost 1 million fragments is what I expect. What's that going to look like on a gel? It's not going to look like discrete bands like we've been looking at today. Instead, it's going to look like this big schmear. Okay? Maybe the brightest part of the schmear is the most abundant sizes. Some sizes are quite small, some sizes are very big. But that's the kind of thing that we expect. If we want to find any particular gene in this array of the whole genome, we're going to have to do some special work to find the one gene we want to study. We didn't have to struggle to find what we wanted to study on a plasmid. There wasn't much there to begin with. But in the whole genome, we have to find a way of zeroing in on what we're interested in studying. We can't really deal with the whole thing at once. Again, another example of a labeled gel. Base pairs, units on the side, the numbers lined up to indicate the size standard. All of this is mouse genomic DNA, so that applies to both of this. This one's uncut, this one's exposed to EcoR1. There's the 1 kb ladder. That style of naming things is not so cryptic, and I think we want things to be readily understandable. Okay, new material finally. Whew, what do we got? 10 minutes left? Good God. This is a diagram from the book that shows double-stranded DNA. The yellow strand and the red strand are complements held together by hydrogen bonds. The blue strand and the green strand are complements. Must be a different sequence held together by hydrogen bonds. If we denature these double-stranded DNAs by boiling, usually we take a test tube of DNA and we boil it. We can render all the DNAs in that test tube single-stranded. The other thing we can do if we don't want to add heat, we add sodium hydroxide. So an alkali solution will also make DNA go single-stranded. And sometimes you'll see that's what's done instead of boiling, if boiling is inconvenient. We don't put acid up against the DNA because that degrades it. So that's why we don't do that particular insult. But whatever insult we do, if we do heat and then we slowly cool, the wonderful thing is, again, diffusion, moving things around in that liquid solution, the complements will come back together all by themselves without any smart person with eyeballs deciding they belong together. They come back together spontaneously owing to the laws of physics. It may take a long time. It may take weeks or months if what we've taken is the whole human genome and boiled it and then we expect it to find itself again. That's going to take a lot of time. Sometimes we design experiments differently, that we boil and separate very short double-stranded molecules, maybe only 50 bases long. They will snap back together quite quickly, especially if they're highly concentrated. So concentration affects the speed, and the complexity of the DNA affects the speed. But the understanding is what you denature, you can renature. Heat to break the hydrogen bonds, cool, and they'll come back together. If you've used sodium hydroxide, alkali, to break the hydrogen bonds, over here we would say neutralize. So you go back to a pH of 7 and things will just come back together. Now, there's a lot of biological processes that depend upon separating and then reannealing. I suppose replication, transcription, translation all involve that. But certainly laboratory techniques where we really do boil things. Many laboratory techniques just depend upon that one little physical property. And there's almost always, remember the other day we said there's always a question about base content. There's always a question about purines and pyrimidines. There's always a question about temperature. At what temperature do things denature? This is an interesting question because it really accomplishes two things in terms of testing your understanding. Which would denature first? Double-stranded DNA with 25% C, double-stranded DNA with 15% T. So this question has you do the base content thing already. 25% C. What if I just convert that into a different thing? What if I said, well, there's an equal amount of C and G. There's 25% both. 50% of this molecule is GC. That's molecule A. Can I get molecule B in the same units? If it's 15% T, then it's 30% A plus T, and the leftovers is going to be GC, right? So it'll be 70% GC. And so now I've done the base content trick to get these apples and oranges into one same style unit. So far, so good? So that's one goal of that question. The second goal of the question is which would denature first as temperature increases? The implication is we're starting cool and then we're gradually getting hot. GC pairs have 3 hydrogen bonds. So the more GC pairs, the tighter the double strands are going to be held together. This has more GC, therefore more hydrogen bonds. As temperature goes up, this one falls apart first, and it takes a little bit more heat to make this one fall apart. This one will fall apart second. And so being able to relate or determine, like we did here, it doesn't come out and tell you it's about hydrogen bonds, but that's what it's about. So knowing At2GC3 will allow you to solve the required question on melting temperature. There's always a required question on melting temperature. Here's a question from a 201 exam about this concept. It's taken from your book, so I think this is exactly in your book. If you raise the temperature of a solution containing the following 3 DNA molecules, in which order do you suppose they would melt? This question is different from my percent GC. The percent approach assumes that we're talking about molecules that are really big or at least as big as each other. This question's different, where we have a really short molecule and somewhat longer molecules, and yeah, there's different amounts of GC there. What you're actually supposed to do to solve this question is literally count the hydrogen bonds. So you can count up all the GC pairs. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, 13, 14, 15, 16, 17, 18, 19, 20, 21, 22 if I've done it right. Whoops. So A's got 22 GC pairs. That would be times 3. That makes 66 hydrogen bonds. How many AT pairs? 1, 2, 3, 4, 5, 6. 6 AT pairs. That makes times 2. That makes 12. There's a total of 78 if I've done it right, H bonds. And so I want to do the same thing for each of these molecules. In a small molecule, you can't do it for percent. You have to count literally the number of hydrogen bonds. And then again say, the implication is we're starting with a low temperature, and the one with the least hydrogen bonds would break apart first. It may well be the small one, but I don't want to take the time to count it all up. Should we take the time to count it all up? I'm bored. Okay, no one's voting. I'm going to move on. Okay. Something you can do with this idea of DNA hybridization once again. There's this thing, I think it's Kathy's favorite word, is probe. So a probe is often defined as a short, single-stranded DNA, like the one floating in the breeze up here, to which we have added something we can see. Sometimes that something is a radioactive phosphate. Radioactive phosphates are easy to add to the 5' end of any nucleotide, so sometimes it's radioactive. Radiation gives you cancer, so we tended to avoid that. Sometimes you stick something that glows a pretty color out there. Glow-in-the-dark molecules can be stuck at the 5' or the 3' end. I think I've just stuck something out there. Now my single-stranded DNA is a probe because I can see it, because of that little label that I put out there. Maybe that's the other important word, label. Some sort of a label. Some people put a little chunk of iron out there and then they can find it again with a magnet. There's a bunch of different ways to go. The glow-in-the-dark stuff's pretty good. So this question is, given the single-stranded target down below, where would the single-stranded probe stick? This is a case, a lot of times students know that they're looking for the complements of that sequence. What a lot of students don't realize is we have to make an anti-parallel pair. They can't just be complementary. Notice how I've got the 3' end here. I've also got the 3' end there. We're going to have to flip this guy over so that we are making a complement which is also anti-parallel. So I've told you, high school students, we assume they know all about complementarity. They get that right every time. None of them know about polarity. So most of our questions are about polarity. Maintain the anti-parallel and complementary structure. And so that's where this probe would go. So if your question was, gee, where can I find that sequence somewhere else in a more complicated situation, like in a chromosome or in a gel? If we wanted to find a particular human DNA sequence in a gel, we could take a probe and we could see where it hybridizes, where it would like to form hydrogen bonds. Hybridization of labeled DNA probes to chromosomes in situ. A chromosome in situ, if you take a cell and you break it open, the chromosomes are in there. If you throw away the broken down cell membrane and you throw away the cytoplasm, you can often get those chromosomes in situ as they are, be a duplicated chromosome, and you can get it to stick to a microscope slide. And I guess for a human being, you could get 46 duplicated chromosomes to stick to a microscope slide. You could dip that slide in sodium hydroxide, make all those chromosomes as they are. That's what in situ means. We haven't rendered them a chemical. They're still in the shape of a chromosome, wrapped around that protein scaffold as chromosomes are. If we stick them to glass, stick it in alkali, make those targets now go single-stranded, we can apply a fluorescent probe to that metaphase spread, and that is called FISH, fluorescence in situ hybridization. And you end up with pictures that look like this. Here are 2 copies of some chromosome. These are duplicated chromosomes. That's 2 identical sister chromosomes held together with a centromere. And I guess this has been probed with 1, 2, 3, 4, 5, 6 different probes, each of them attached to a different colored fluorophore. So it's a kind of a real pretty experiment. If you wanted to know the order of these 6 genes on this chromosome, there's your answer, and you get the answer in like 10 minutes. So what is this really a picture of? There is a double-stranded DNA. There is its exact copy, a product of replication, those 2 identical sister chromatids held together with a centromere. That's what this is. I can kind of see a centromere right there, a little pinch. I think that's what's going on. This is the homolog. So this chromosome came from mom's side of the family, this chromosome came from dad's side of the family. This is after replication, so we have a double-stranded DNA and then another double-stranded DNA, 2 identical sister chromatids held together with the centromere. Now that's all vocabulary from an earlier course. Homolog, chromatid, chromosome, centromere. So yeah, you were supposed to learn those terms. Stuck to the glass, exposed to sodium hydroxide so they can't move far from the glass, but I think we've exposed all those hydrogen bonds. And then you throw the probe in, and I've got that golden labeled probe sticking to its anti-parallel complement there and there and there and there. Remember, my probe is a single-stranded molecule, so it's not going to stick to both strands, it's only going to stick to that way. And so I think that's why we have two bands of hybridization there, two bands of hybridization there, because we really have four chromatids, because we've set these two homologs side by side. So this is the kind of molecular experiment you can do just with a single-stranded DNA probe. Next, we're going to do something with probes that's a little more complicated. So I think we'll stop. We'll talk about a southern blot, and then we'll talk about some more modern twists on the southern blot. In previous semesters, we've kind of done a southern blot. Kathy's asked me to stop doing southern blots because they're so old, but we will still teach them, but we'll do something else in our laboratory next week. So all of these slides are posted and more. We'll get through the rest of these on Friday. Certainly, if you've worked on your lab report and you have questions, again, it's a good time to maybe bring them up.